Unlike many people in the room, so I'm not a spine surgeon and don't really work on spine cancer. So I am going to present a bit of maybe a little bit of an outsider perspective, or talk a little bit more from a molecular side. So I'm a computational biologist, as was alluded to. So we do a lot with big data, molecular information, trying to predict, prevent, treat disease. And so that's what I'll talk to you about today. And I will range a little bit broadly in topics to try to bring out certain concepts, which I hope will be relevant and interesting to people here. Okay, obligatory disclosures, consult for a number of companies, also founder of a company called Aravale, which I'll talk about a little bit today, and they did help fund one of the studies, the Pioneer 100, that I'll talk about a little bit. Okay, so as was mentioned, I come from a place called the Institute for Systems Biology. Just to give you a little background, ISB has recently become an affiliate of Providence, which Swedish Hospitals is as well. And so this is a big 50-hospital system. Lee Hood, who is our president. If you don't know Lee Hood, Lee was the inventor of the automated DNA sequencer, one of the initiators of the Human Genome Project, member of all three national academies, one of, I think, 7 people living that's true of, and so forth. Anyway, so Lee has now become the chief science officer for the Providence system, and ISB has become now a research arm for Providence in part. And so we're very interested in sort of going from basic science all the way to clinical impact, and that's a big centerpiece of what we're trying to do. So let's start with the problem. So here's a slide. This is taken from Nick Shork, who we've been interfacing with a lot lately. And he wrote this paper for Nature called Imprecision Medicine. And if you're not familiar with this, what is shown here is you see all the figures. In green are shown the people who actually responded to the drug. And in blue are shown people who did not benefit from the drug. These are the top 10 selling drugs in the United States. And so if you look at this, Abilify, Nexify, Humira, so forth, what you'll see is that it ranges from the pretty good, where Abilify, where you've got 1 in 5 patients who take the drug actually benefit from it, to say Nexium, where it's about 1 in 25 patients who actually benefit from taking the drug. This, of course, has led to all kinds of interest in developing precision medicine, which is the right drug to the right patient at the right time, where we understand better how to understand in advance who's going to benefit from this and who not. And of course, we could get into all kinds of interesting discussions about the economics of figuring this out and who wants to find this out and who doesn't want to find this out and so forth. If we think back to cancer, of course, there was a big molecular effort for the Cancer Genome Atlas, or TCGA. The TCGA, one of the big data centers for the TCGA actually resided at the Institute for Systems Biology. Ilya Shmulevich there was one of the PIs and one of the co-chairs for the Cancer. So a lot of this analysis that goes across all cancers happened at ISB in under Ilya's leadership. But basically here, what we've seen is that cancers, as I think everyone is aware, they have a lot of heterogeneity. And this represents both a challenge and an opportunity, and a big potential for personalization and trying to map this heterogeneity back to that imprecision medicine problem I talked about on the last slide. There's also many different actionable driver events, and as people are thinking about personalization in cancer, of course, the notion is that we shouldn't think so much about the site of origin, right, breast cancer, prostate cancer, brain cancer, et cetera, but really about what are the molecular drivers of the disease. Is this an EGFR mutated cancer? Is it a KRAS mutated cancer, and so forth? And there's a lot of interest in that. That's logistically challenging in some ways, just because most medical departments, medical fields are historically organized around sites of location, and there are some important reasons for that. And certainly in surgery, that makes a ton of sense. But at the molecular level, there's this trying to rethink how do we think about getting a better treatment outcome for cancers more generally. Let's see if we can get back to the slide set. There we go. Right, next. Okay, so again, obvious advantages then to personalized therapy, which is to try to take out of this milieu of everybody that you're seeing a subset of who will actually respond, who will be non-responders, and also trying to identify things like adverse effects. We're in discussions with a number of pharma companies right now, and many of them have problems with this, pretty serious problems, where they have a drug. There's one company we're talking to pretty soon. They have a drug. It works for about, or it works or doesn't have a negative effect for about 99% of people, but in 1% of cases, there's this kind of horrible side effect to it, and it's bad enough in 2 of these cases that the drug is mostly not given by doctors out of this fear that for 1% of patients they'll have these really negative adverse effects. So there's a big interest, obviously, in also being able to have molecular markers that will target who that set of, or at least who are high risk for being in that very adverse set, so you can carve them out, allowing the drug to then help the people that it can help. And in terms of targeted therapies, as people are probably aware, there have been a number of targeted therapies that have come out. Gleevec was one of the really big successes. Herceptin, another really big success. But there's many of these targeted therapies coming. Of course, they never come as fast as we hope, and there's a big push to try to figure out ways to accelerate this process. All right, so all of this has led, of course, to a big push towards precision medicine. President Obama gave the impetus for the Precision Medicine Initiative, which is an initiative that's funded at about, $150 million or so dollars put in for building out a big cohort. Eventually, they want to do genomes of 1 million people, along with clinical records and so forth, to try to drive interest in that area. And so these things are often called moonshots. This is going to be a little bit of an aside. It's sort of interesting. There's the cancer moonshot from Joe Biden, right, which is about $1 billion that they are saying that they're going to put into cancer research. It was pointed out recently, and I think it's quite interesting, that the actual moonshot was $147 billion a year in order to do that in the 1960s. And so when we talk about moonshots today for disease, there's nothing like the investment that was actually put into, say, the actual moonshot. But anyway, that's an aside. So from our neck of the woods, and this is a terminology coined by Lee Hood, but basically we think a lot about P4 medicine, which is a little bit more than personalized medicine or precision medicine. P4 stands for predictive, preventive, personalized, and participatory. And I'm actually going to talk through all 4 of those as we go through the talk. But we see this type of medicine as being essentially inevitable, and it's really driven by 4 big mega trends in society that are forcing this. So one is the advent, we would say, of systems biology, systems medicine, the idea that you can take genomes, proteomes, metabolomes, all these kind of huge amount of information and cross a threshold from ignorance to knowledge on the way that a lot of the systems of the body are working from a broad molecular level. The digital revolution, so wearable devices, kind of interconnectivity, ability to get measurements of many different kinds, big data and analytics, which allows us to then analyze this data, and then patient-activated social networks, which are really, we think, key to patients sort of advocating for the same kind of personalization technology application in medicine that they get in other areas of their lives and so forth. All right, so how do we get to personalization? So I'm going to deviate a little bit now and tell you a little about what we're doing, because I wanted to set the stage for discussing how we get to personalization in a deep molecular way. So I want to talk just very briefly about determinants of health in the United States. This comes from a 2007 New England Journal of Medicine paper, and it's been roughly estimated that over a person's lifetime, about 30% of their lifetime health is attributable back to their genetics. About 60% is attributable back to behavior and lifestyle, and only about 10% of a person's lifetime health is attributable back directly to the healthcare system. So one of the things that this motivates is us to think a lot about that other 90%, the genetics, the behavior, the environment, all those things in a more holistic way. And I think that medicine is starting to move that way in terms of embracing a broader range of what's happening, say, in a person's life to have that impact in health. So this has led us to think a lot about an area that we call scientific wellness. And we believe, well, if you look at the U.S. healthcare system today, we spend about $4 trillion in the United States on healthcare, as I'm sure everyone here knows, which is close to 20% GDP. So that's a big spend. There's also, according to National Academy of Medicine report, 97% of those dollars are spent on care after illness. Okay, so this is all, and people in this room very aware of this, I'm sure, deal with these kind of acute problems all the time. There's also, of course, a wellness industry that exists now, some of which is fantastic, some of which is, say, dubious. And so there's a lot of discussion. There's a lot of things out there that don't have a very deep scientific basis. So what we see emerging now is an area that we call scientific wellness, which is in essence taking all of these, the science, the new ability to make tons of measurements, to validate, to have feedback for the individual in a way that will allow us to have a new industry that we call scientific wellness that differentiates itself somewhat from traditional healthcare in the sense that it's focused on trying to optimize wellness and predict and prevent disease before it happens rather than waiting till after some sort of catastrophic event. And so we think that this is going to grow. Lee likes to say that this will be bigger than the healthcare industry in 10 or 15 years. So anyway, so to make that a little more concrete, what Lee Hood and I announced in 2014 was a project that we called the 100K Wellness Project, or the 100,000 Person Wellness Project. And so, and this was put into various publications. But basically, the idea here was that we really wanted to make an impact on the ability to predict and prevent disease. And so what we announced was an intention to recruit 100,000 individuals, do whole genome sequencing on everybody, and then multiple times per year measure thousands of different analytes out of the blood, proteins, metabolites, clinical chemistries, do gut microbiomes, wearable devices, and in essence build these all into what we call dense dynamic personal data clouds. And the big goal of that was to create a data set that was dense enough, so we had enough kinds of information, dynamic in the sense that we could follow people over time, so we could start to understand the early warning signs for the major human diseases, so we could develop ways to predict them and prevent them. And so that was kind of the big vision of starting this up. And so as we did that, what that turned into, in a sense, was a company that we ended up spinning out called Aravale. And so Aravale is a company, it's focused on scientific wellness, so it tries to provide actionable information from these data clouds to individuals so that they can optimize their health in the moment. And then as a sort of a byproduct of this, what you get is this longitudinal data. So if people are happy and delighted with what they get from that experience, hopefully they'll stay with us. And if they check a box saying that their data can be used anonymously for research, then we can mine that to try to understand the early warning signs for disease, metrics for wellness, and so forth. And so far, we're very grateful for this. 95% of people who have come through the company so far have agreed to do this. And actually, Providence itself has now put 2,000 of its own employees into this program as well. So that's being a test bed for us to figure out how do you impact healthcare with these new kinds of data. And we've raised just under $40 million for the company so far. So this scientific wellness that we're pushing, and I'm going to bring this back to cancer here in just a moment, we're pushing in two different directions. So Aravale, as I mentioned, is the main way that people come in as from a consumer side. But then also within ISB Providence, what we're able to do is use these dense dynamic data clouds as part of clinical trials so that we get a lot of information from certain disease populations. And we're also able to launch trials around findings that come out of surveying, in the broad population, interesting relationships so that we can then translate that to diagnostics, therapeutics, and so forth. Okay, so all of this came out of an initial study that Lee Hood and I led in starting in 2014 called the Pioneer 100. And basically this was to prototype the scientific wellness approach I just mentioned in 108 people. And so, and this is all being published, I should say. It's going to be in Nature Biotechnology, coming out in June, I believe. So here, so we did whole genome sequencing on everybody, as I mentioned. So that gives you millions of differences between individuals just in terms of SNPs, single nucleotide differences, and so forth. For labs, we did about 700 different metabolites, about 400 proteins, about 150 different clinical labs. That was done 3 times. We did gut microbiome 3 times a year. And then we also did self-tracking devices. So what this does for us then is it gives us a database of actionable possibilities. This is growing all the time. And then we're able to create these dense dynamic personal data clouds on individuals. We had great health outcomes. We were able to reverse some diseases in this population. I won't go into that in the interest of time, but I do want to show you just a little bit of the data. And so here, this is just a circle diagram. Every blue line here represents a correlation we were able to identify that crossed different data sets. So how is something in your microbiome related to something in your blood? How is your metabolite related to your protein? How is your protein related to your genetic risk for disease, and so forth? And what I want you to consider is that, so there's 4,000 correlations here. No one had ever before measured all of these things from the same set of individuals ever. So what that means is that this is a way for us to start looking at relationships of what's happening in the body in a way that just wasn't possible before. We didn't have the measurements and the computation and all the different things necessary to do this. This, by the way, is just a small tip of an enormous iceberg. I mean, this is a small sample size at the moment, right? This is only 108 people. We now have more like 1,500. But it's just an ability to start understanding how these things are interconnected. And I'll give just a very abbreviated version of this. So one of the things you can do with that big correlation network then is you can break it down into modules, and you can start to see which modules move together, which, what's really related to what. Some of this, of course, is understood from, all the background literature, but we can do a lot of this de novo now. And so you get all kinds of different relationships. One in particular that I'll show here is this one right here, which says inflammatory bowel disease, and it's connected to cystine, okay, and these networks. And I'm skipping all the details of how we actually generate these networks in the interest of time. But that's interesting. So inflammatory bowel disease here doesn't actually mean that somebody had inflammatory bowel disease. Okay, so no one in the study has IBD. But what it means is that this is actually the genetic risk for inflammatory bowel disease. So we can now take all these genetic risk scores, and there's genetic risk scores of pretty high quality for about 60 to 70 different diseases now. And what we can do is look at what are all the different analytes that are related to that genetic risk. And so in this case, IBD connects with cystine. And I'm just going to explain that, not because we're focused on IBD today, but because I just want to explain a concept, and then I'll come back to cancer with all these things. So what we were able to find then was that there was a strong negative correlation between the genetic risk that someone has for IBD based on previous GWAS studies and the amount of cystine that's in their plasma. So the lower your plasma, the higher your genetic risk for IBD, or really causality would be opposite direction. So we then looked into the literature, and it turns out this is pretty interesting. So for one, there was a study where they did a look at just a normal case control study in blood of people that have IBD, the disease, and those who don't. Turns out plasma cystine comes up as one of the top candidate biomarkers for IBD out of the blood. We didn't know that going in. Very interesting. For ulcerative colitis, right, which is a form of IBD, the lower your plasma cysteine is a dimer form of cysteine, but the lower your plasma cysteine, turns out the higher the clinical activity index, the worse you are. So that was interesting. So we find this relationship, but remember, we find it in a population in which nobody has the disease, just looking at the impact of their genetics. So there's two really important there's two important implications from that. One is that you can disambiguate differences that probably predate the disease from those that are related to the onset of the disease. In other words, and remember, the people in our study, they range in the age from 20s all the way to 88 plus. I say it in that weird way because our IRB, you can't give ages out over 88. Anyway, so you've got this huge age span, and yet we already find it. So what does that mean? That means that the kind of data we're talking about can be used as a correction factor on essentially any case control study in medicine. So if you're doing a case control study, right, we and anybody else do the same thing. You get a bunch of people and they have, you match them for age and sex and smoking status or whatever, and you have your cases and your controls. And then you look at all the differences between the cases and the controls. But it stands to reason that the people that got the disease at the same age as the people that didn't were at higher genetic risk for that disease. And what this enables you to do is to build background models of all the differences that you expect as a function of that difference in genetic risk, and not as a function of the onset of the disease. So it helps you figure out what are related to risk factors and what in fact are might be better as diagnostics of the onset of a disease. So there's different ways to suss that out. The second big implication, which is even more interesting, is if we see how this genetic risk manifests in the body, and that's a big topic for us, the manifestation of genetic risk in the body, then what it tells you is that there may be a way that you can intervene. So here's a hypothesis that comes out of this. Cystine, turns out, sits upstream of glutathione pathways. Glutathione, of course, what combats oxidative stress in the body. Oxidative stress, a big trigger for IBD. So, immediate hypothesis, you have these genetics. They lead to a less effective glutathione pathway via this suppressed cystine. In fact, that link has been shown in another paper as well. So you have that depressed ability to combat oxidative stress. That means over the decades of your life, you're marginally more likely to transition into a point where you're outside of the boundaries of what you can control, and you'll trigger into that disease. Okay, so it comes up to a very simple hypothesis. And what's even more exciting is if that hypothesis is true, it means that you can take somebody who is at high genetic risk for IBD and by fairly simple type of nutritional interventions, you could potentially change them to being at low risk for IBD. And what we want to do is to just do this for every disease we possibly can and just map out, here's the genetic risk, here's how the genetic risk manifests in the body, and here is how you might be able to intervene so that you make it less likely to transition into those disease states in the first place. Okay, so that's where we're going with that. Okay, the other thing, even more directly, that we're interested in is monitoring for when people unfortunately do transition into disease. Right, doing this program, it's not like, we're not ending disease, it's still happening. I get that question every once in a while, which is kind of blows my mind. But basically, so we have transitions from people. So this is, so Aravel's been around, as I mentioned, for about 18 months now. We've got about 1,500 people who have come through a substantial part of the program, a few thousand more who have signed up, and basically who are starting the program. But already we've started to see transition to disease in a few cases, unfortunately. But let's just look at that. So one of we call our participants the pioneers here. So one of the pioneers was diagnosed with chronic leukemia. So this was a person came through the program. So they had these 4 historical blood draws, 2 microbiome samples. And so what we were able to do, and this just shows in each of these cases where they were, but they were just a massive outlier in the population in terms of just the number of leukocytes. So this is just screening, just seeing what there were problems. At Aravel, of course, not a medical place. It's a wellness-focused company, so this gets referred back to the healthcare system, back to places here like Swedish. And essentially, what we found was that this really high elevated leukocyte level did in fact mean this person had leukemia. They came back to the medical system, were diagnosed with that, are now getting treated for that properly. And all that showed up in the blood, though, well before any particular symptoms. So we do see things like that. Perhaps maybe a more interesting example, although this is this is very bad cancer, obviously. We had a pioneer that was diagnosed with stage 4 pancreatic cancer, and according to Bert Vogelstein and others, when you're diagnosed with pancreatic cancer, it's estimated that you've probably had that disease for about 5 years. And so when this person got diagnosed, they had been in the program for about 18 months. And so we went back and looked in some of the discovery assays we do, the proteomics and so forth, that aren't understood very well yet. But one interesting thing popped out of here, and these are small n, right? This is going to become big N, but I'm just giving you the first inklings of this. So here's a particular protein. It's in the Notch signaling pathway. It's a regulator of cell growth. It's made heavily in the pancreas and so forth. And it turns out if you look at this distribution, this is the distribution of the level of this protein for every person that's in the Aravale cohort. And what you'll see is way out there to the right, there's a major outlier. And it turns out that this is the woman that got pancreatic cancer. Now, it's a different question of whether the next time we see this, that means pancreatic cancer, right? There's a lot of work that goes into this, but I just want to give you the taste of where we're going. So what we're building are methods that look at outlier detection, and we do outlier detection across everybody. And this is a very different approach than like a classical biomarker type approach. So with the biomarker type approach, we'd make the assumption that this particular protein, that this is the signal, right, when we try to develop a biomarker around this. And that may turn out to be true. But what we're actually doing and what we believe in more is that we'll do an outlier analysis. So we'll look at how many cases do you have something that's just really outside of the norm. And then what you do is you do a computational analysis across all those outliers to see if there's a particular pattern to them. Right? Do a bunch of these come from the pancreas? All right, maybe something's happening there. Do a bunch of these come from the brain? Maybe that's where they're happening, and so forth. And so what this does is this gives us an ability to then dive in and try to understand for every person, like, what is this predictive form of disease? And it gets into massive personalization, because then you're actually analyzing the totality of what's really unusual that's happening in a person's body that's different from just, this is the biomarker for this disease. That lets us get into really personalized, even N of 1 type analyses of individuals in terms of what's happening in their body. And this is just the very first inklings of that. We have about a dozen or so cases now of people who have transitioned to disease where we can go back and identify something that was unusual. And that will get, of course, massively better as we go forward. And then that's ultimately what we want to turn this into, is an ability to really monitor for disease in a major way. And we're trying to figure out ways we can partner with Providence and with other organizations around driving this forward. So this, of course, helps serve as the basis then for a highly personalized, preventive, predictive, participatory type medicine, the P4 medicine that Lee Hood's talked about for a long time. So we want to be able to look at wellness, understand wellness better. I was on a panel recently with former chair of Harvard Medical School, Denny Ausiello, and it was really interesting because he had a statement, and I really liked it, so I've used it a number of times. And he says, medicine is the only discipline that doesn't study its own gold standard, which is wellness. And I thought that was really, really an interesting statement from somebody like him. And so we have wellness. So we want to understand wellness, and we want to understand what the earliest disease transitions look like and how reversible they are. With 10,000 dense dynamic data clouds, we think we're going to be able to start identifying a number of these transition points for the most common human diseases. We're going to generate blood diagnostic markers for the earliest stage of each of these diseases. We want to use our systems-based approach to understand the early disease perturbed networks. And again, can we intervene in them? That's what we really care about. This can lead to different drug target candidates, drug combinations, all kinds of different things like that can be interesting. And the idea is to have a drug that will actually reverse the disease, fix the network, so forth, before the disease happens, right? So we're trying to strap Humpty Dumpty in rather than waiting for him to fall and then try to put the pieces back together. That's our mindset primarily. And we think that this will be the basis for preventive medicine in the 21st century, is this kind of broad approach. All right, so now I'm going to shift gears a little bit, again, just to end in the last 10 minutes or so that we've got, and just say a few words about how we can use some of these molecular data types. So there's, of course, a ton of signals that exist. We believe very strongly that blood is a very powerful window into health and disease. And this is really turning, this is really medicine as an information science, right? Which is not sufficient in and of itself, obviously, to the actual delivery of medicine, but we think it's an important component. But how do we read these signals? Signals in the body you can start with a massive complexity, you can get down to simplicity. This was a paper I did several years ago in PNAS, but in that case, we worked with MD Anderson Cancer Center on trying to come up with a marker that would differentiate gastrointestinal stromal tumors from leiomyosarcomas. They used to be classified as the same cancer about 20 years ago. Then people realized they were different. They have very differential responses to Gleevec, for example. And what we found here was we measured 43,000 different transcripts, and but basically through a bunch of machine learning algorithms found a very simple pattern. It turned out there were these two genes, one called obscurin, the one at that time didn't even have a name, it was called chromosome 9 or 65, now called prun2. But basically, if the expression of obscurin was higher than the expression of prun2, it was a GIST, and if it was the opposite, it was an LMS. And that turned out, at least in our study, to be true like 98% of the time. And so it came out as a very simple, very easy to apply diagnostic for a very simple question. And I just give that as an example because those are, of course, there's tons and tons of those things in medicine, and what we want to do is to be able to understand patterns that are useful to that. So that was a particular clean one that came out. Another one, just to give, this is a paper in Cell Systems with Lee Hood and with Charles Cobbs, who's a surgeon here, brain cancer surgeon here at Swedish. This paper came out last month. Anyway, this is looking at a signature for glioblastoma made out of proteins that exist in the membrane of glioblastoma. It turns out 4 of these are secreted into the blood, and they appear at pretty massively different levels in the blood of someone, say, that has glioblastoma versus normal controls. I won't go so far as to say biomarker yet, so we have to do a lot more validation, but it's a pretty interesting process where we were able to come up with a set of proteins that are very sort of characteristic of the tumor, a subset of which you can actually observe in the blood. So we think this is a promising avenue to follow up on. You don't have to look at these in isolation. You can also think of them in network context. There's lots of networks, and my group and others do a ton of work around metabolic networks and gene regulatory networks in particular, also signaling networks. But basically, you can utilize these to dive in and try to understand signatures from a network perspective. So DRAC is a method that we developed. I won't go through the details of it in the interest of time, but basically it looks at the rank ordering of the components of a particular network from highest concentration to lowest, and uses that as a proxy for the state of the network in interesting ways. And it turns out you can do some interesting math here, develop a template ranking for any particular phenotype, and then you can do comparisons to that quite easily to come up with network perturbations that are characteristic of a disease. So one example of this would be in prostate cancer. It turns out the MAP kinase network, if you apply this kind of approach, you can pick up a lot of interesting signals. I won't go through that in the interest of time, but just to say in terms of accuracies, for example, if all we know is the expression level of the 15 genes that are in the MAP kinase network, we can differentiate normal prostate from prostate cancer, from metastatic prostate cancer, about 95% accuracy, based only on those 15 genes and the rank order in their expression. So there's all kinds of interesting things you can do with these types of approaches. I will say that there are major barriers for clinical use. There was a National Academy of Medicine study, then the Institute of Medicine, they changed their name, but the National Academy of Medicine, and I was actually a member of this committee, so co-wrote this report, but basically this was around trying to address a few big things, which is one, why are there 1,000 papers that say they've got biomarkers and only 2 are approved every year? And also this also reviewed anyway, a quite famous case at Duke, which was an issue back at that time. But basically, what we needed to do here then is that I just wanted to bring this up because there are all kinds of ways computationally, and you see these signals, and there's all kinds of ways that they degrade. And it becomes a really important systems problem. And I don't mean systems just like my kind of computation. I mean systems in terms of like the whole medical system, how we set up, exactly how the measurements are made, exactly how it's transferred, how it works in one hospital versus another, and so forth. But there's all kinds of pitfalls here that you can fall into that has something that seems to be an amazing signal at one place that then degrades to nothing in another place. Part of it is because the transcriptome or the proteome or whatever is responsive to everything in the environment, right? Not just whatever we have in our head. So there's all kinds of factors that matter, and so that data reproducibility becomes absolutely the key factor. And so there are a lot of issues, and if you want to understand that in detail, I do recommend the report. It goes through this whole process in a lot of detail. But what I wanted to say, though, is that we do believe that you can get around these challenges. And in fact, we have now been able to get 2 multi-parameter proteomic measures clinically approved. So one of these is from something called Integrated Diagnostics, or INDI. It's a spin-out company from ISB. Lee and I and others were co-authors on this paper. But basically, this was the first test. And what it does is it takes a 13-protein marker. You run a lot various analytics across it. But basically, what you're able to do with it is to differentiate somebody where you have a lung nodule, and you're wondering, is this cancerous? Is it not? And what it actually does is it can for a subset of individuals, it can rule out that they have a cancer. And so that, and the estimate on that is that it would save, in terms of not doing the surgeries, that would save the healthcare system approximately $3.5 billion per year by deploying the tests to be able to rule out some of those people that wouldn't benefit from this. And I could go into that in a lot more detail, but not here. And then the second I'll mention, Lee and I both are advisors for another company called Sera Prognostics. And Sera Prognostics is a company, they've just done the same thing, a multi-parameter blood test, in fact, and we just wrote, along with the chief scientists from both those companies, Lee and I just wrote an article that kind of goes through the unified process. Both of these companies use essentially the same pipeline to get to the end product, and in both cases resulted in a clinically approved test. I think so far the only 2 mass spec, multiple, multi-parameter protein omics tests that have made it to the clinic. So I just wanted to point that out. So there's huge obstacles. We think we've figured out a lot of ways to get around them, and we're actually having clinical success in getting these things out the door. I mention that also because with our Providence colleagues, we're very interested in finding clinical problems that really matter, for which these kind of predictors would be useful, and developing approaches to get there, just like we have helped with doing in these 2 cases. All right, so to sum up, dense dynamic personal data clouds, we think this is going to be a big impact. We're going from very little information comparatively about patients to a very lot of information. That's going to be really the foundation for personalized medicine going forward. And we think that these clouds also need to be deployed very broadly in clinical trials. And we could go into that in more detail, although I won't here in the interest of time. So just to conclude, we do believe that these types of systems approaches, the dense dynamic data clouds, will be important for informing prevention, and I gave some examples of that. For diagnostics, I gave some examples of that. For therapeutics, which I didn't really get into too much today, but we could go into that, drug target identification and so forth, as well as recovery. And I'll just mention briefly, just like we did with the Pioneer 100, we're now launching a program with Swedish Cancer Institute, where we're looking at cancer survivors and applying a scientific wellness approach to try to get people back to recovery following chemotherapy, radiation, and things like that. So we do have a whole program that we're developing there that I didn't get into today. With that, let me thank the great members of my lab, people that contributed to work I talked about today, James Eddy, John Earls, who did a lot of the analysis for the Pioneer 100, as did Andrew Magis, and of course, big thanks to Lee, and then other people that from Swedish, Charles Cobbs, Tom Brown, and others that we work with a lot. And thanks.